Welcome to Biology with Deepika, and in this video we will learn about bacteria, its structure, its general characters, plant characters of bacteria, its shape, its motility factor, and lot more. So let's see all this and elaborate this. The first is bacteria are microscopic unicellular plants without chlorophyll that reproduce by fission. They are microscopic unicellular plants without chlorophyll. They don't have chlorophyll that reproduce by fission. Antony von Leeuwenhoek in 1675 discovered bacteria from polluted pond water and tartar scraped from the teeth and are called wild animalcules. This discovery was published in his book The Secret of Nature, discovered by Leeuwenhoek. Linus in 1758 placed them in the class Vermis and called them chaos. The term bacteria was coined by Ehrenberg. This is very important in point of view of exam. The term bacteria was coined by Ehrenberg. The bacteria have been variously called fission fungi and schizomycetes. Varro called the bacteria Animalia minuta. Burgee placed the bacteria in Prosophyta group and wrote a book, Manual of Determinative Microbiology. This book is known as Bible of bacterial classification. Oswald Tipo placed the bacteria in division Schizomycophyta of Thallophyta, while Carl Nageli placed them in Schizomycetes. Whitaker placed all prokaryotes in kingdom Monera. Lister developed culture technique and sterilization technique. He discovered the antiseptic nature of carbolic acid and proposed antiseptic theory. Louis Pasteur is known as father of microbiology. He proposed the germ theory and called the bacteria germ. This is very important in point of view of exam. Louis Pasteur is known as father of microbiology and he proposed the germ theory. Robert Koch, father of microbial technique, discovered the causative agents of anthrax, which is caused by Bacillus anthracis, tuberculosis, and cholera. Robert Koch gave some rules to prove that the bacteria are the cause of disease, the rules known as Koch postulates. Koch postulate doesn't applicable on obligate parasites. This is very important. Koch postulate doesn't applicable on obligate parasites. For example, Mycobacterium lepra. Koch obtained pure culture of bacteria firstly. Then we will go see how the general characters of bacteria. The basically bacteria are unicellular. Cell wall of bacteria is made up of murein or peptidoglycan. This is important. Well, it doesn't have a well organized nucleus, Golgi complex, mitochondria, and other membrane bound cell organelles. They are smallest of all living organisms with a cellular organization. Mostly the bacteria are heterotrophic due to absence of chlorophyll, but some bacteria are photoautotrophic or chemoautotrophic. We will see about it in later videos, in my upcoming videos. Major reproductive method of bacteria is binary fission. The plant characters of bacteria are presence of cell wall, which is made up of acetylglucosamine. Bacteria can synthesize enzyme and vitamins. The food is absorbed in solution form and can synthesize the food also. When we go to size of bacteria, bacteria are small and their size generally ranges from zero point 0.1 to 1.5 micrometer in diameter and 2 to 10 micrometer in length. These are very important in point of view of exams. The smallest bacteria is Haemophilus influenza. The longest or largest bacteria is Epilepsicum fischelsoni, and the largest or longest filamentous bacteria is Beggiatoa mirabilis. These three are very important. Then we will move to the shape of bacteria. Here we see in the diagram how the shape of bacteria look like, and we will see in details. These are classified into 5 types by Kohn. A scientist Kohn classified the shape of bacteria into 5 types. The first type is coccus. This is the coccus type, circular like looking. The coccus bacteria are spherical, highly resistant, and smallest bacteria depending upon the number in the group they may be. They are smallest bacteria and they are spherical. The first one is monococcus. Monococcus, it is a single sphere. Here we see in the diagram monococcus. For example, micrococcus and dialyster pneumocystis. The second type is diplococcus. Diplococcus is paired. Here we see in the diagram, they are paired, attached, 1, 2. And for example, we can see this in Diplococcus pneumonia. The third type is Tetracoccus. Group of 4 cocci are present. For example, in Micrococcus luteis, here we see tetrads. And Streptococcus, it is chained. For example, we can see this in Streptococcus lactis. Here, Streptococcus, it is present in chain. We can see this in Streptococcus lactis. Then Sarcina, it is 8 to 64 or more bacteria are found in cubical mass form. Here we can see Sarcina ship. 8 to 64 or more bacteria are found in cubical mass form. For example, Sarcina. Then Staphylococcus found in irregular bunch. Here we see it is irregular bunch like coccus. It is Staphylococcus, and we can see this in Staphylococcus alvis. This coccus type, and it is single sphere. Diplococcus is paired. All this is important to remember. The second type is Bacillus. They are rod-shaped bacteria. These are the common type of bacteria, Bacillus type rod-shaped bacteria. These are the rod-shaped bacteria. The single bacillus, only one rod-shaped bacterium. For example, E. coli or Lactobacillus here, one single shape. The Diplococcus are paired. They are paired. Here we see paired. These are paired. We can see this in Diplobacillus. Then Streptobacillus. It is chained. Here we can see Streptobacillus. It is Bacillus anthracis is its example, and palisade bacillus is another type. A matchsticks-like arrangement will be present. For example, Corynebacterium diphtheria. These are the type of Bacillus bacterium, Bacillus bacteria shape. The third type is Spirillum. They are spirally coiled bacteria. Here we see Spirillum shape. They are spirally coiled bacterium. For example, we can see this in Spirillum volutent and spirochete. The fourth type is Vibrio. It is comma shaped. Here we see in the diagram. And the fifth type is pleomorphic bacteria. They can exist in different morphological form at different times. For example, Rhizobium. It can be found in 3 forms, X, Y, and Z. They are pleomorphic. They have different morphological forms. So this is all about the shape of bacteria. Then we will move on to motility factor in bacteria. Bacteria is motile as well as non-motile. Movement in bacteria takes place by means of flagella. We see all those here. This is flagella, this is flagella. Bacteria move by means of flagella. Depending upon the number of flagella and where they are present, bacteria are of following types. Number 1 type is Atricus. The flagella is absent. Here we see Atricus. For example, in Micrococcus. The second type is Monotrichus. Only one flagellum is present here. For example, in Vibrio and Pseudomonas. The third type is Cephalotrichus. Here we see the flagellum, a bunch of flagellum is present in one side or one end of bacteria. For example, we can see this in Pseudomonas fluorescens. The fourth type is Lophotrichus. When a bunch of flagella is present on both sides of bacterium, this is Lophotrichus. Here we see on both sides of bacteria, bunch of flagella is present. For example, we can see this in Salmonella. The fifth type is Amphitrichus. Here, single flagella is present in both sides of bacterium. Here we see in the diagram Amphitrichus, how a single flagellum is present in both sides of bacteria. For example, Spirillum. The sixth type is Peritrichus. Flagella is found on the whole body of bacteria. For example, in E. coli, like this type. So this is about motility in bacteria. How flagella help, number of flagella can helps in classifying the bacteria as Atricus, Monotrichus, Cephalotrichus, Lophotrichus, Amphitrichus, and Peritrichus. This name and the distribution of bacteria is very important in point of view of exam. Then we will move on to the structure of bacteria. The cell structure of bacteria is studied as mentioned below. The number one is capsule or the slime layer. This is the capsule as we see in the diagram. It is a, or we call the slime layer here. This is the capsule. Some bacterial cells are covered by gelatinous covering. When the gelatinous covering doesn't form a persistent layer but present as a loose mass around the bacterial cell is known as slime layer. When the gelatinous covering form a well-defined persistent layer, it is called capsule or glycocalyx. This layer, outer layer. It is made up of polysaccharides, dextran, leaven, and cellulose. It prevents the bacterial cell against desiccation, maintain viscosity, and prevent the attachment of bacteriophage. The next we will move on to the plasma membrane of bacteria. Here this is the plasma membrane. It represents the outermost membrane of cytoplast. The term cell membrane was coined by Nageli and Kramer. This is very important in point of view of exam. The term cell membrane was coined by Nageli and Kramer. It is made up of lipoprotein and semi-permeable, and it is semi-permeable in nature. The plasma membrane consists of a continuous layer of phospholipid molecule in which globular proteins are embedded. The globular proteins are of two types, extrinsic proteins and intrinsic proteins. The mesosomes, which we see here near the plasma membrane, this green color, it is in all gram-positive bacteria. The plasma membrane forms invaginations called mesosomes. It is found in all gram-positive bacteria or chondroids, the bacterial mitochondria. Mesosomes help in DNA replication, septum formation, and it stores respiratory enzymes. This mesosomes is very important. Then we will move on to the cell wall. It is the outer covering of cell and provides shape and rigidity to the cell. The cell wall is like this. Here it is the outer covering of the cell and provides shape and rigidity to the cell. The bacterial cell is made up of mainly peptidoglycan or murine, which is a type of mucopeptide. It is found only in prokaryotes. Here we see the how it is arranged. I will explain you in detail now. The peptidoglycan consists of two components, a peptide protein, which is composed of amino acids connected by peptide linkages, and a glycan or the sugar portion. The glycan portion forms the backbone of peptidoglycan. It is composed of alternating units of amino sugar N-acetylglucosamine. This is N-acetylglucosamine, the red color, and the green color is N-acetylmuramic acid. They are joined together by beta-1,4 linkages. How they are joined? These are two amino acids and they are joined by beta-1,4 linkages. The peptidoglycan chains are laterally linked by short chains of 4 amino acids which are attached to N-acetylmuramic acid. They are laterally attached by 4 amino acids which is attached to N-acetylmuramic acid. Here. The 4 amino acids of this tetrapeptide are L-alanine, D-glutamic acid, L-lysine in gram-positive bacteria, and diaminopimelic acid in gram-negative bacteria, and D-alanine. These are the 4 amino acids, L-alanine, D-glutamic acid, L-lysine, and D-alanine. The tetrapeptide chain are also interlinked by a peptide bridge between the carboxyl group of an amino acid in one tetrapeptide chain and the amino group of another tetrapeptide chain. A peptide bridge is also present here. The cross linkages can occur between tetrapeptides in different chains. As a result, the peptidoglycan forms a rigid multilayered sheet. As there are cross linkages present, between tetrapeptides here, cross linkages are present between tetrapeptides. Peptidoglycan form a rigid multilayered sheet. The cell wall of gram-positive bacteria is single-layered and thick. It is made up of only peptidoglycan, comprises 90% of the cell. Lipids are also present but in less quantity. So this is all about the cell wall of bacteria. Here we see another important thing to mention. Here we see the N-acetylglucosamine and N-acetylmuramic acid. They are linked by beta-1,4 linkages. This is very important. And It is important to understand this picture also. The next we will move on to the difference between gram-positive and gram-negative bacteria. The thickness of the cell wall of gram-positive bacteria is 150 to 200 angstrom, and the thickness of the gram-negative cell wall of gram-negative bacteria is 75 to 120 angstrom. The cell wall in gram-positive is single-layered, smooth, and in contact with cell membrane. The cell wall is two-layered and in contact with cell membrane at few places. The lipid content in the cell in gram-positive bacteria is 2 to 4%, and the lipid content in gram-negative bacteria is 20 to 30%. The amino sugar are 10 to 20%, and the amino sugar in gram-negative is 2 to 8%. Muramic acid content is 70 to 95% in gram-positive bacteria, but it is very less in gram-negative bacteria, which is 5 to 20%. Mesosomes is common in gram-positive bacteria. Mesosomes is less prominent in gram-negative bacteria. Pili is rare in gram-positive bacteria. Pili is common in gram-negative bacteria. Teichoic acid is present in gram-positive bacteria, but teichoic acid is absent in gram-negative bacteria. Gram-positive bacteria may produce exotoxin, but gram-negative bacteria may produce endotoxin. Porine is a protein which is absent in the cell wall of gram-positive bacteria, but porine is present in the cell wall of gram-negative bacteria. There are few pathogenic forms found in the gram-positive bacteria, for example, Bacillus and Clostridium and so many others, but pathogenic forms are common in gram-negative bacteria, for example, E. coli, Rhizobium, etc. Then we will move on to the flagella and what is sexpeli. The cell of bacteria have several hair-like structure on their cell wall called called flagella. Here we see this is pili, and this is the flagella. The flagella is composed of fibrin protein and called flagellin. The flagellin is similar to keratin in hairs and myosin. Flagella has 9+2 arrangement. This is very important to remember. In addition, minute hair-like pili are also present in male cell of E. coli. Each pilus is made up of pilin protein. It is a contractile protein. When pili help in conjugation, it is known as sex pili, and when it helps in attachment, it's called fibri. It is the same pili, but when it helps in conjugation, it is known as sex pili. When it helps in attachment, it is called fibri. The next we will move on to the nucleoid or nuclear body. Here we see in the diagram, this is the nucleoid or nucleoid. It has no nuclear membrane, hence called nucleoid or nuclear body. The genetic material is known as genophore or bacterial DNA. It lacks the histone protein. The nucleoid is single, circular, and double-stranded DNA molecule. The nucleoid is circular. E. coli gene is 1 mm large, having 10,000 genes. The number of supercoiling varies from 12 to 82. This is very important to remember about E. coli. Then we'll move on to plasmid and episomes. Bacteria may possess extracellular genetic material called plasmid. If plasmid has 9 genes, it has self-replicating capacity. Then we will see what are F plasmid or sex plasmid. It is a conjugating fertility factor which are required to pass the genetic information from donor to recipient during conjugation. The cell carrying F factor acts as a male while the cell lacking it acts as a female. The male cell bears sex pili. The R plasmid. It is the resistance factor. It carries genes for resistance to antibiotics and other antibacterial drugs. The Col factor is the factor that converts bacteria to killer strain or poisonous and are known as calcinogenic factor. The episomes remain attached to the plasma membrane and are larger than the main chromosome, about 1 cm in diameter. They get easily incorporated to the main chromosome of nucleoid region. So this is about flagella, sex pili, nucleoid and plasmids. Then we'll move on to few questions. In all of my videos, I I make some questions at the end of my video, important for any exams. The first question is germ theory of disease was given by Louis Pasteur. The second question is bacteria are considered to be plants because they have rigid cell wall. The third question is the general shape of bacteria is rod shaped. The fourth question is bacteria which are smallest in size are corky. The fifth question is the circular DNA is seen in bacteria, that nucleoid we have studied now. The sixth question is the structure formed by bacterial genome is generally called nucleoid. The seventh question is diaminopimelic acid and muramic acid are constituents of the cell wall of bacteria. And the eighth question is bacteria have the following organelle, we can say mesosomes. So these are few questions from my video. So this is all about bacteria from this video. So please like, share and comment if you like this video, and please subscribe for my upcoming videos. Thank you.